Hi, everyone. It's Dr. B here, and this is Physical Principles of Quantum Biology, Chapter 3, where we'll be defining non-triviality for quantum biology. Let's begin. First of all, I'd like to say that definitions matter. They matter in physics, they matter in law, and in practically every area of our lives where we use words. The definitions of those words have meaning. It's interesting in quantum biology where the word trivial has often been used in debates about the nature of quantum effects in biology. And the thing is that the word trivial hasn't always been clearly defined in these debates. Some people have talked about trivial effects referring to entanglement or coherence in systems, quantum coherence, but other definitions have been used even to say that something is only non-trivial quantum mechanically if you need to take graduate courses in quantum mechanics to understand it. So this kind of inconsistency or even ambiguity has in some respects hindered scientific progress in quantum biology. Although it's been the subject of a good debate, it has downplayed the importance of quantum coherence and entanglement in biological systems and ultimately obscured the fundamental role of quantum mechanics in determining many aspects of macromolecular, that is biomolecular structure. So if we want to really understand what's going on, we need to know what we really mean when we say something is non-trivial. And a principled definition will align quantum biology with established many-body physics, and it will improve cross-disciplinary communication, providing a suitable physical basis for modeling biomolecular structure and function. So, trivial versus non-trivial. What ideas have been developed so far? I've put a few references here at the bottom you can read through. First of all, if we think about trivial in the most colloquial sense, it means something is negligible or irrelevant. Something in physics often is stationary. Of course, your dynamics are trivial if you're sitting still, you're not moving. And in biological systems, as I mentioned before, this concept of triviality in the colloquial sense can be entirely subjective. Now, in physics, trivial does have a formal meaning, and in fact, it can refer to interacting many-body system, a system with multiple physical particles that can be reduced to a non-interacting one. And the properties of this many-body system can then be reproduced by an ensemble of one-body problems. And those of you who are familiar with statistical mechanics might be aware of the microcanonical ensemble, which is the case when we have many systems that don't interact. The statistical average of those systems then simply becomes our average energy or gives properties like the temperature of our system. But the important thing is that the system becomes trivial when we have particles that do not interact. Now, the simple idea is that if you don't have interactions between your particles, there's not much physics you can do. It's like just having a baseball floating in space. There's really not much that's going to happen. So something becomes non-trivial when it has irreducible interactions or correlations between particles that are needed to reproduce observable phenomena. And thus the so-called interaction kernel, the part of the equation that accounts for those interactions, cannot simply be set to zero. So getting back to quantum mechanics, when is entanglement trivial? Entanglement is this spooky action at a distance, as Einstein called it. It gives the impression that two or more particles can interact with each other even when they're very far apart. What's really going on is that they are correlated, and they have correlations that cannot be rationalized using classical physics. They seem to defy the laws of classical physics or laws of classical reasoning being correlated in ways that our usual logical way of viewing the world could not describe. So electrons, for example, or any quantum mechanical particle, particles that are identical, every electron is in fact identical to every other electron, are formally entangled simply due to the fact that they're literally indistinguishable. You can't tell the difference between one electron and another electron, and in this sense, you can never separate the identity of one electron from, in fact, every other electron in the universe. But if electrons are far apart, then there's no observable consequences to the inherent entanglement between them, and this entanglement can be identified as trivial. This is a phenomenon called cluster separability, and you can look it up in Asher-Perez's famous quantum mechanical textbook. What happens, though, is once the wave functions of electrons begin to overlap, then you can no longer treat them as if they were separable. In fact, this is the basis for chemical bonding, which requires wave function overlap and results in entanglement. In fact, even the simplest molecule, like the hydrogen molecule, has inherent quantum effects that still cannot be defined using density functional theory if you want to study any of the dynamics of the system. We understand how the hydrogen molecule works when it's simply sitting with the two hydrogen atoms that comprise it are sitting at their equilibrium distance. But in fact, if you now simply stretch the bond of a hydrogen molecule, there are no known density functional theory approximations. This is the main workhorse of chemistry that will actually adequately describe that system. And so even a very simple molecule like the hydrogen molecule has non-trivial entanglement in this sense. So ultimately, if we want to do quantum chemistry, let alone quantum biology, we need to think about how we're going to encode these quantum mechanical correlations, which typically are encoded in what's called the exchange correlation potential in, for example, our density functional theory model of the system. So, if we want to think about what triviality means in this formal sense I mentioned before, this is known as Landau triviality. So if the electrons don't interact, we can imagine that they're separable, and in fact an interacting model becomes trivial if there's an equivalent. It's called free field theory, which can be considered in high energy or the short distance limit, for example. This is the exemplary case when the system properties reduce to those of a non-interacting system. And if the system of interacting particles can be equally described with a system of non-interacting ones, well, then we say the interactions and, in fact, all the interesting physics of the system go away. It is a trivial system. So the criterion, formally speaking, in quantum field theory, and I would say quantum mechanics more generally, is that a system is said to be non-trivial when the interaction terms are not negligible, when we need to account for these complex interactions in order to reproduce our experimental results, in order to make predictions ultimately. So that's very interesting when we apply it to something like density functional theory, as I mentioned, this workhorse of quantum chemistry, because in fact, density functional theory is based on the very same idea. The idea of mapping an interacting system of electrons to a non-interacting one. So in density functional theory, we imagine we have, of course, we have some chemical system where the electrons are surrounding the nuclei of atoms and they form bonds and perhaps undergo chemical dynamics. But we replace all this system of interacting electrons with an imaginary, a fictitious system of non-interacting electrons, and thus we have, a very trivial system. But if we now introduce an effective potential, what would be basically a classical potential, we can now imagine we can reproduce all the effects of quantum mechanics with that potential. And in other areas, this term would be called the interaction kernel. It's the term in our expression that accounts for all of the interactions between the electrons in a singular way. Now, the Kohn-Sham equations of quantum chemistry are exact in principle. We have theorems that tell us that if we can write down the equation for a non-interacting system that looks just like an interacting system, it looks like it insofar it has the same density of electrons, then in fact it will have the same energy and in fact all of the same properties of our true interacting system. So in fact, Kohn-Sham density functional theory already gives us a simple way to see whether the interactions, the quantum mechanics of the system, are trivial. That if we can just ignore this exchange correlation function, then we no longer have any interesting dynamics. We can call it a trivial system. But of course, you may have guessed, or if you're familiar with quantum chemistry, you know well, that understanding and developing model approximations for this exchange correlation term in our Hamiltonian, in our energy function for the system, is really a central focus of quantum chemistry. And there is still, as of yet, no ultimate exchange correlation function that simply works for all systems. So it's a bit tricky. This tricky part is known as the inverse density functional theory problem. That if we knew what the exchange correlation function was, then we could use this sort of semi-classical non-interaction picture to reproduce all of the complex correlations of quantum mechanics, but we don't know what it is. And so a lot of work has been done to ultimately identify how to take the density of electrons of a system that we know, say we got the density of electrons of a molecule from X-ray crystallography or some other experiment, then really the question is, how would we use that to determine what this magical seeming exchange correlation function, this potential term, actually is? Well, as of yet, there's no clear way to do that. Quantum chemistry is not solved. And in fact, we have to come up with approximate ways to account for these quantum mechanical interactions, these quantum mechanical correlations. And it's tricky because the simplest approaches, the mean field or local density approximation approaches, they ignore dynamic correlations between the electrons, dynamic fluctuations. They miss out on long-range correlations between the electrons. They struggle even with static correlation, although that might not be surprising because that's where entanglement comes in. And they cannot capture so-called dispersion forces that are universally found in aqueous molecular systems. These kind of van der Waals interactions and London dispersion forces sometimes can be treated with classical approximations, but often cannot and require full many-body quantum mechanical treatments. And so approximating the exact many-body densities is as difficult as some of the hardest efficiently verifiable problems in quantum computing. This is not a trivial task by any means. And imagining that we can simply make these quantum mechanical interactions go away, trivializing the problem by rolling all the tricky quantum mechanical stuff into this effective classical approximation that we can treat adiabatically, have someone suggested, really just trades one problem for another, but does not make the actual difficulty of the problem trivial in any meaningful sense of the word. So what we actually have in quantum chemistry and biochemistry and the whole structure of matter are a hierarchy of complexity of approximations. At the very lowest level of our pyramid of complexity, we have classical force fields where we still aren't truly classical because we need our parameters for our kind of elastic approximations for our bond stretching and torsional angles, all the molecular vibrations that we put into our classical simulations. All of these have to be put in by hand, and these parameters ultimately for the various bond stretching and so forth effects come from quantum mechanics. We either need to determine them from experiment empirically, or we need to do a quantum mechanical calculation somewhere in order to figure out what the parameters should be. Now, these still don't account for many-body correlation or non-locality effects, or at best account for them only partially. And then we need to, at that point, resort to what are called mean field quantum mechanical methods, where we treat these quantum effects using semi-local or weak quantum mechanical correlations. And this is the kind of thing that comes out of what's called Hartree-Fock theory, local density or generalized gradient approximations, which begin to account for weak correlations in our system, but are fairly modest. Then we scale up to these meta generalized gradient approximations, meta GGA, hybrid, double hybrid, more complicated methods that have been put in still essentially by hand in order to account for exchange and correlations effects. Some aspects are ab initio from first principles, but ultimately a lot of work has to be done, essentially by hand in order to get the approximation to work for one system. And as of yet, no approximation works for all systems. You kind of have to choose your system and choose what modeling method works best for that type of system. So there's no single solution to all forms of chemical systems. Now, as we go up our hierarchy of complexity, now we can look at truly non-local correlations, which can begin to capture medium-range quantum mechanical effects. This is things like the random phase approximation and many-body dispersion correction methods, before we really go beyond what is even known in density functional theory today, and we have to go to post-Hartree-Fock or so-called wave function methods, perturbation theory, coupled cluster theory, multi-reference configuration interaction, very advanced stuff. And frankly, it doesn't even stop there. If you look at the reference I've put on this slide, you can see there's an enormous amount of work to understand these kind of correlated materials, strongly correlated quantum effects that we see, yes, even in biology, in iron-sulfur clusters or other transition metal complexes that are frequently, in fact, ubiquitously found in living systems. So there's, roughly speaking, two types of quantum correlations to consider. Static correlations that we can treat adiabatically. That's a stationary approximation where we imagine the nuclei sit still and the electrons whiz around so fast they're just a blur. And these kind of stationary systems still can involve non-local quantum effects that are kind of embedded. So you don't imagine, the molecule in your mind going through any kind of strange non-local quantum effects. If you have a picture of it or a model of it, it just looks like a density of positive and negative charges. But in fact, in order to account for the exact characteristics of that density of electrons in space, typically there's still non-local quantum correlations that can involve entanglement, either weakly or strongly so. And now, on top of these static quantum correlations, there also can be dynamic quantum correlations, even for a system that appears to be sitting still, because, of course, our atomic and molecular theory of matter doesn't really allow for anything to ultimately sit still. We have the uncertainty principle that requires there are fundamental fluctuations, fundamental dynamics, even for a system which is sitting as still as possible. And these produce things like van der Waals forces and, as I mentioned, dispersion interactions in a system. So predicting the quantum dynamics of the nuclei and electrons remains one of the greatest challenges today in computational chemistry. At the lowest order, what's called the zero order, we have this Born-Oppenheimer approximation where we imagine the nuclei are sitting still. They're as if they were clamped in space. And we have then first order approximations from Fermi's golden rule, where we have simple tunneling events or quantum transitions, quantum jumps, if you will, that we can define using seemingly classical transition rates that, again, still must be calculated using quantum mechanics. And finally, what people tend to think more of when they think of, non-trivial, difficult to calculate, difficult to model systems are these higher order correlation effects that are challenging and can be challenging not just for coherent systems, but for decohering systems, because often those decoherence effects, relaxation effects, are a result of higher order interaction effects between the electrons and nuclei. So none of this is really trivial, and it certainly gets difficult very quickly for complex systems, photoactive systems that are undergoing ultra-fast dynamics. So how do we deal with this? So if we start in our quasi-static, as we imagine it's nearly static, sitting still kind of picture. The conventional approach is that we compute our quantum mechanical properties once, we parameterize our semi-classical models, and then we run classical dynamics, and perhaps we introduce some quantum jump effects into the system with a stochastic kind of equation. And this is very practical and often can be predictive at molecular length scales and times, but they hide all the non-local correlations inside our fitted parameters. They mask the reality of these nuclear electronic vibronic couplings, and it breaks down when we have significant non-adiabatic quantum effects that cannot simply be accounted for with these simple, semi-classical or first-order quantum mechanical approximations. So this is very nice for, pumping out lots of simulations, but don't necessarily get down to the heart of some of these dynamical enzyme functional effects that we see certainly in photoactivated enzymes and also in redox enzymes. It's very challenging. So how do we account for non-trivial dynamics in electron and proton transfer systems? So tunneling mediated reactions, either electron tunneling or proton tunneling reactions, often can involve non-trivial dynamics that require at least a first order Fermi's golden rule type effect, and sometimes higher order effects as well that go beyond simple surface hopping. We imagine one electronic surface when the nuclei sitting in it, and then an electronic transition, a quantum jump or a tunneling event takes place, and it hops to another so-called adiabatic surface potential function in which the nuclei sit, which again has all of the inherent difficulties of any quantum chemical calculation with exchange correlation effects and whatnot. So this is really the basis for a lot of biochemistry, the basis for understanding life itself, because all of life, from photosynthesis to respiration, begins with these electron transfer and often for many enzyme-mediated processes, also proton transfer effects. So we should not think that the system, just because it appears stationary, is scientifically trivial, because, as I mentioned, many electrostatic problems that appear to be trivial actually define computational grand challenges simply to account for the many-body electronic correlations in the systems. So the difficulty is physical, not merely numerical. Sometimes it's hard even to know where to start in writing down our equation, let alone solving it on a computer. And accurate energies and structures can result from these irreducible quantum correlations that cannot be simplified to non-interacting models. And so even static effects require this many-body quantum treatment. I cannot stress that any more strongly than I have. I would therefore say that structural quantum biology is a core non-trivial subfield that underpins all resulting downstream dynamics of your non-equilibrium living system. If you just want to take a couple molecules, even the caffeine molecule is an example of a molecule that people building quantum computers today would like to be able to simulate from first principles. But the size of the molecule, which is big but not incredibly huge, not like the size of enzymes and macromolecules, actually, if you wanted to simulate it from first principles, you would really need a quantum computer that's beyond what we even can do on the best prototype systems developed by commercial and research laboratories alike. And if we want to go to something like the nitrogenase enzyme, the nitrogen fixing enzyme, it's so important for agriculture. It's loaded with iron sulfur clusters and is far beyond the limits of both classical and quantum computing systems today. In order to even begin to tackle these things, we have to go through enormous rigmarole of elaborate quantum mechanical approximations just to get the system, a representation of the system in a form that we can work with at all. So the ordering of quantum biological phenomena by complexity is very important to define rigorously. We don't want to have one person using one definition of trivial and another person using another arbitrary definition of trivial, whether you went to grad school or not. We really want to lay things out in an ordered series of approximation from least to most complex. The least complex being mean field, semi-local, or simple adiabatic approximations. As we increase complexity, we include dispersion and weak non-local correlations. These can be often accounted for in advanced density functional theory methods to good order, but they get a little more complicated as we begin to introduce open quantum system effects, decoherence effects, and then ultimately the most complex systems are those that include strong static correlations, complex vibronic couplings, or quantum electrodynamic effects, either simply in the form of the absorption or emission of individual photons, or potentially even cavity quantum electrodynamics effects like you would see in a quantum optics lab. So if we want to think about the criteria we have for defining what is trivial or non-trivial and have a ladder of complexity that we can really use in our understanding how to do simulations of quantum effects in biology, first of all, we start with this idea that if there is no need to include quantum interactions at all between the particles, then it's truly trivial system. We have something called a free electron gas. And this is very good for understanding the band structure of materials, but it's really just the beginning of our approximations before we begin to introduce electron interactions and correlations in either a weakly or strongly interacting way. If we have weak interactions, then perturbative or simple or relatively simple density functional theory approximations can work, and we don't need to worry about the really thorny quantum mechanical correlations that emerge in these so-called multi-reference models, where we don't just start with a singular equation, Slater determinant is called. We have to go to a very complex system of equations with multiple terms interacting with these coherent effects. Then we next need to consider dynamical criteria. If we look at the next level, simply analyzing how complex our system is when it's sitting still is just the first step because, of course, things move. We have quasi-static approximations in most of chemistry, in transition state theory, for example, where we simply consider the system in terms of effectively stationary or very slow-moving nuclei that ultimately cannot become entangled with the electrons that are quickly moving in the system. But as we relax that, we go to these first and higher order non-adiabatic effects, they're called, and this ultimately can become quite difficult to solve very quickly. And how difficult it is really is an area for analyzing already today in terms of quantum complexity theory. There's an enormous amount of work that's been done in complexity theory and in computational theory and quantum computing to understand the complexity of systems, when we can model it efficiently classically and when not. There's still actually a lot of discussion going on to ask how big an advantage a quantum computer should be able to give for doing quantum chemistry, because in principle it should be a very large advantage, but so many very good heuristics and approximations have been developed for quantum chemistry that it's still not always entirely clear the scope of the advantage you can get from doing something brute force on a quantum computer versus being very thoughtful about it in terms of developing your heuristic approximations for a classical set of computations instead. So there's an enormous amount of work to be done here. What would then be the best practices? First of all, we need to be clear in our notion of triviality, and we should define it the way computer scientists do, or the way they've defined these things in quantum field theory, in order to really give a clear picture of where the interactions go from non-interacting to weakly interacting to strongly correlated systems. We can then think of these things in terms of quantum chemistry approximations, local density approximations, hybrid approximations, or coupled clusters, singles, doubles, even triple kind of interactions. And we want to specify very clearly which correlations, static, dynamic, and so forth, are being included or omitted. And then we can justify our approximations. We want to justify the Born-Oppenheimer or quasi-static simplifications that we make and test for our system's sensitivity to non-adiabatic or kind of dispersion effects, fluctuation effects, and identify where fitted parameters, even in our semi-classical models, truly are representing fundamental quantum mechanical effects and quantum correlations. So in the broader context, quantum biology still goes beyond even quantum chemistry. Quantum chemistry gives us the electronic and molecular properties of these systems, but of course living systems interact with light through vision, photosynthesis, even vitamin D production, and so there's a lot of light matter interactions in living systems and even photonics. Crystals are very commonly found in the living world, and crystals with photonic properties are well known in biological systems. Living systems are open quantum systems, so we have to be concerned with decoherence and not just thermal effects, but dynamical relaxation effects that often are not trivial to calculate at all, even as the system decoheres from a complex quantum state to what you would think is a more simple one. Often the Hamiltonian for the system, particularly if it's strongly correlated, still relaxes to a state which is very hard to solve computationally. And we ultimately need an integrated framework that accounts for all of these things in order to give a full description of all the processes, a multi-scale description of quantum effects in living systems. So in conclusion, I'd like to say that this idea of trivial as simple structural or stationary is misleading, that quantum effects pervade biomolecular structure and function, and that formal model-based definitions reveal that even static structure can reflect highly non-local correlated quantum effects. So we need to not just debate about whether we think it's trivial or not, subjectively or even informally, but really establish a very rigorous, clearly defined, ordered ladder of complexity in order to define the simplest to the most complex quantum effects in biochemistry and quantum biology more broadly. Ultimately, the structural aspect of quantum biology is indispensable because it lays the foundation for all other non-trivial dynamical quantum effects in living systems. So thank you very much for your attention today. If you've enjoyed this lecture, don't hesitate to download your guide to quantum biology, Physical Principles of Quantum Biology. We've reviewed Chapter 3 here today. It's available for researchers, students, and anyone who's curious to venture beyond the classical explanations for biological systems. And finally, if you'd like to know more, you can always contact me at my email address and continue watching these video lectures on YouTube. Have a great day.